All right. Hello, everyone. Is anybody there? Hello. Ayan, ayan, ayan. Nag-ingay na silang lahat. Hello. Hi. Hi, everyone. All right, so I'm back. Okay, so we're now going to talk about anemia of chronic disease. And this is another important part of the exam. And that's the reason why I put it here also again. And I think they're going to ask this on clinical chemistry, and they might ask this on clinical chemistry as well as immunology. But for the discussion on hematology, I'm just gonna jot it down for you guys a little bit quickly. So the main cause of anemia of chronic disease is actually the inflammatory process itself. You see, the body can't distinguish between inflammation and inflammation and infection. So it blocks the transport of iron in the bloodstream. So it has something to do with iron metabolism as well. All right. Now, anemia of chronic disease is the most common in patients with the following disorders. Or is that one? Kidney disease, autoimmunity, and rheumatologic disorders. So, what do I mean by rheumatologic disorders? This could be either rheumatoid arthritis or anything related to the problems in the connective tissues. Autoimmunity, there's wide range of those ones. But you just need to remember the mnemonic CAR. Okay? So, when you have anemia of chronic disease, just remember the mnemonic CAR. It's spelled with the K. So, kidney diseases, autoimmune disease, or autoimmunity, and rheumatologic disorders. All right. Anemia of chronic disease is the most common form of anemia in what type of settings or environment? So, it's basically seen in patients who are hospitalized. So, in a sense, it's an iatrogenic form of anemia. Iatrogenic meaning that it is hospital acquired rather than uh, rather than it is acquired from, say, for example, an infectious disease, or it could be drug induced. Okay, so the more the patients are hospitalized and the more treatments they undergo, the more likely they are to develop inflammatory disorder, this an inflammatory process. And that's the reason why we need to we need to also monitor patients in case of in these cases, because they might suffer from anemia. All right. So anemia of chronic disease is the most common anemia with patients of what psychologic disorder or psychiatric disorder. These are patients who are alcoholics. Okay. So alcoholism is considered as what? It is considered as as a psychiatric disorder under the DSM-4. What's the DSM-4? It's the Bible of psychology. Okay, so no further discussions on that. Why? Because when you ingest alcohol at constant rate on a daily basis, it produces an inflammatory response. Why? Because the alcohol is not part of ethanol is not part of the human body. So therefore the body would recognize it as something as foreign, and it would mount an immune response in the form of inflammation. So that is going to release a particular enzyme with a particular disease molecule, and we're going to talk about that one later. All right, what is the most this one what is the most important molecule in case of anemia of chronic disease? This is what we call hepcidin. Hepcidin, sorry, hepcidin. And the organ that produces it, from the name itself, hepa, the liver, and it is important in iron regulation. What do we mean by iron regulation? Well, it basically dictates whether or not a um, particular instance in a particular event in the human body requires more iron or decreased iron. How does it know? Well, the body is designed in such a way that it will be able to recognize the need for iron. In cases of blood loss, hepcidin will decrease in production. All right? How does it do that? The relationship of it is that it is inversely proportional. Okay, the more serum iron is present, the more hepcidin you have, the less hepcidin you have. Okay, because the more serum you have, the higher the hepcidin, because you want the body to produce more, to get more iron, either in the form of the diet or in the storage form or in the diet. Okay, what is the storage form again? It is ferritin. Where with hepcidin act on that one? It will act on the hemosiderin stores in the body. By the way, guys, what is hemosiderin? Hemosiderin is actually your ferritin in a form of like this. The body will go the how iron is metabolized is like this. This is your this is a enterocyte. Okay, enterocyte are cells that are found in the gut or the small intestines. This is your enterocyte. It's the nucleus. Let's pretend it's the nucleus. There's receptors here, receptors there, an enzyme there. Okay, there's another receptor here. Sorry, it's a transport a molecule. A transport molecule is here. An enzyme is here. Iron will go from the diet through this particular part, but it is in the ferric state. Okay, the one on this side, the enzyme here will convert it into ferrous iron to become ferric state, and it will pass through this particular entry. This is what you call ferroportin. These two are ferroportin. So it goes in and it goes out. Hepcidin will block this one so that iron will iron in the bloodstream will not go out. All right? Hepcidin will also block it here so that iron will go in. Okay? So that's the action of hepcidin. Hepcidin will also block surface molecules in the sorry it will also block surface molecules in your liver. It doesn't look like a liver, but but that's it. Why? Because the liver is also a source of hemosiderin. Okay? So inside the human body, inside the enterocyte, you have what you call apoferritin. Apoferritin will bind to the iron molecules to form ferritin. It has two fates. It will go out or it will stay inside as hemosiderin. Okay? It gets stored as hemosiderin. If it goes out, it will go to apotransferrin. It will bind to what you call an apotransferrin. And when iron binds to it, it will become transferrin. All right? So that's basically what happens to iron. So why is it important? Because hepcidin will block the ferroportin surface molecules so that iron won't go in the circulation. All right? That's why it's inversely proportional. So what receptors are targeted by hepcidin? As I mentioned before, ferroportin in the duodenal cell, which prevents transport. Anemia arises because of ineffective erythropoiesis due to the abnormality in iron metabolism. See what happens? Your body keeps on releasing hepcidin in times of infection. But as I mentioned before, the body cannot recognize okay, the body cannot recognize whether an infection whether a particular event in the body is an infection or an inflammatory process, and therefore it will release hepcidin. Once it releases hepcidin, it will block the action of ferroportin. It closes the ferroportin channels. Once it closes the ferroportin channels, a patient will suffer from anemia. Why? Because iron is important in normal erythropoiesis. Without iron in normal erythropoiesis, the cells will become microcytic hypochromic anemia. All right? That's the reason why I discussed the iron metabolism in the body. Okay, so hepcidin blocks the action of ferroportin. True or false? All right, this is a true or false question. One way of the body's one way the body protects itself is by releasing hepcidin. That is, of course, true. We've answered that earlier before. Why? Because iron is actually an important part of microorganism survival, of microbial survival rather. Why? Because without iron, without iron, certain metabolic processes in some bacteria is inhibited, as well as in humans as well. So the body itself has a design or a mechanism that would say, I don't want to share my iron with microbes. Therefore, I will release hepcidin. You understand the reason why hepcidin is important? Because we're not the body is not the body is not sharing the iron to the microorganism. So it's trying to prevent its multiplication or growth by inhibiting the body by inhibiting iron storage and iron reabsorption or iron absorption. So microorganisms will die. All right. So how do we differentiate anemia of chronic disease versus anemia associated rather or with IDA? Well, there are four things that you have to consider. Again, we have to consider iron binding capacity, storage iron, and serum iron. What do I mean by percent saturation? This is the amount of iron that is saturated in the serum. It's rarely tested in the laboratory, but the only thing that you need to look out for is these three again. Why are these three important? Because these are the questions in board exam. Okay, so they might present a case like this. You need to differentiate whether or not your choice would be iron deficiency anemia, anemia of chronic disease. It's very easy. Serum iron is decreased in this particular case. All right, in both of them. TIBC is high. TIBC is decreased in this particular case. Why is it decreased? Because there are iron, there is no iron entering. Okay? There's no iron entering. So it's increased. It needs more. But what about in anemia of chronic disease? Why is iron binding capacity decreased? Because you still have iron in store, but the reason why you don't have iron in the serum is because hepcidin blocked it. You still have iron inside the body. However, it can't go out, and so TIBC is higher. All right? So TIBC is lower rather. Ferritin is low in patients with iron deficiency anemia because, of course, they don't have iron stored. Anemia of chronic disease, they have iron stored. However, because of the infection or chronic inflammatory process that is blocked by hepcidin, ferritin cannot be transported as transferrin. So they remain in the storage. All right? So what is the clinical findings for anemia of chronic disease? Well, it's actually difficult to say when you do a peripheral blood smear. Some hematology books, it writes it as normocytic hypochromic. Some books, they say microcytic hypochromic. So let's just say it's in between normo and normo hypo. Okay? So we don't get confused. The main thing that you need to be familiar with in terms of anemia of chronic disease is the hepcidin, the action of hepcidin. If you understand the action of hepcidin, you'll be able to understand or even fill out a blank page like this. Okay? Now, we move on to normocytic anemias. So normocytic anemias, these are anemias that are what? Again, the normal MCV. What's the normal MCV? Anybody? Hello? 8. Oh, galit na galit. Yes, galit na galit si Vanessa. Yes, I love it. I love it. Gusto ko yan. Very, very confident when answering the questions. I love it. It means that you're listening. It means that you're participating. And I love it, girl. Anyway, let's continue. Um, Normocytic normochromic anemias. We're going to first talk about paroxysmal nocturnal hemoglobinuria. Okay? So PNH, paroxysmal nocturnal hemoglobinuria. The major defect in patients with paroxysmal nocturnal hemoglobinuria is you have to remember this. It's the PIGA gene. Just remember a pig, which basically means PIGA gene. All right? Sleeping pig. Why do I say sleeping pig? Because it starts with the letter P. It occurs historically at night. So oink oink. Okay? Small circle, big circle. So sleeping pig. That is my mnemonic. All right? Okay? So that is my mnemonic for you guys. Sleeping pig. When you want to remember what is about PNH, just remember the sleeping pig because that is my mnemonic. PIGA gene is the problem. PIGA stands for phosphatidyl inositol glycan class A. That is your PIGA gene. All right? What is the importance of the defective gene? Why is it important? Because the PIGA gene is responsible for coding your CD55 and CD59. CD59 is also known as your decay accelerating factor. CD59 is known as your MIRL or your membrane attack complex inhibitory proteins. Why is these two things important? Let's go back to your immunology discussion. So in immunology, we talk about complements. I will denote it as C apostrophe. Uh, C apostrophe. I want to use a pen. Can I use a pen? Can you guys see my drawing? I don't know if you guys can see my drawing or you can understand my drawing, but hopefully in your notes there, if you're listening, you're going to note it like this. All right. C. That is your complement. What happens with complement? You basically start with QRS, start with C2 convertase, C4 convertase, then you have C5, C3 convertase. After that one, you're going to produce what? You're going to produce your membrane attack complex that basically starts as C5. C5 becomes C5 convertase, blah, blah, blah. And then it becomes your C5, C56789, which is now known as your membrane attack complex or what we call your MAC, right? Basic discussion on immunology. Why is it important that we have CD55 and CD59 on the cells? I'll give you guys an example. Suppose that the body doesn't have these particular CD55 CD markers on the RBC. Okay. Now, normally, if a cell has it, I'm going to make another drawing. Uh, this one. Let me erase this one first. Normally, if the body doesn't have it, if the body has it, sorry. Normally, the body has it, and it will be like, let's go and make this a yellow color so you guys can understand. That's your RBC. It doesn't look circular, but whatever. All right. So central pallor, blah blah blah. All right. CD55 and CD59, or your DAF and CD55 and CD59. So normally the cells will just go and pass by. Complement molecules are present in the serum, as denoted by these small stars that you have here. Because of CD55 and CD59, these molecules won't attach to your cells. But since you have a defect with the PIGA gene, you won't have these two with you. Okay? You won't have these two with you. And your cells actually become bombarded or peppered with complement molecules. And that's what happens in PNH. Because there is a lot of complement molecules that peppers your or covers your RBCs, they have a tendency to activate the classical and the alternative pathway. Okay, so usually what will happen is the cells will lyse in certain conditions, especially when the person is sleeping. Okay, so that's the pathophysiology of paroxysmal nocturnal hemoglobinuria. Okay, somebody open their mic. Somebody wants to share something with me? Hello? Cyrene opened her mic. Do you have anything to say, Cyrene? Cyrene Alvarez. Right. I don't know why she opened her mic, but yeah, we enjoy hearing the sounds of Manila. But anyway, that's the that's the basic pathology or pathophysiology of your paroxysmal nocturnal hemoglobinuria. Girl, girl, who's that? All right, somebody. All right, somebody. Please mute yourself if you're accidentally entered or entered the voice chat. But yeah, anyway, that's PNH. And what are the essential findings of patients with these type of hemolytic anemia? Well, basically, you're going to have patients with pancytopenia, a normal Coombs test, and normocytic normochromic anemia. What do I mean by pancytopenia? Pancytopenia, anybody? What does that mean? You will encounter this word several times. Anybody? Pancytopenia. Guanyin. What do you mean by what? What are those three blood cell types? You're being vague. I want specific answers. Mhm. And all right. Thank you so much. So, negative Coombs test. Negative Coombs test. What do we mean by Coombs test? We actually have two types of Coombs test, direct and indirect. We're gonna discuss this, and since I'm gonna be your blood bank instructor as well, or was I don't know, speaker as well. So we're gonna leave Coombs test uh, as a floating question for you guys. So what is the difference between them? Okay, we don't need to know the exact details. Direct Coombs test detects the in vitro sensitization. Sorry, in vivo sensitization rather. Sorry, in vivo sensitization, in vivo, and then this one is vitro. Okay, so this one we're going to do for paroxysmal nocturnal hemoglobinuria. Why? Because the cells are already covered with the antibodies, or rather complements. They're already covered with complements. While in vitro, what we're detecting is actually the antibodies that are present. So is indirect Coombs test useful here? No. Okay, so what we need is direct Coombs test. We want to attach anti-human globulin on the surface of the red blood cells. Okay, why? Because it targets the complements that are attached. All right, complements aren't able to fix themselves because there's no antibodies present. So you need to attach anti-human globulin first. Okay, that's the reason why we're doing indirect Coombs test. Direct Coombs test. All right, understood? Okay, why normocytic normochromic anemia? Well, basically, there's nothing wrong with the cells. The problem is it's not actually a problem with hematopoiesis or iron or heme group or the hemoglobin itself. The main problem is actually the surface molecules. Okay? So that's the reason why it's normocytic normochromic anemia. It's within the same size and it's not going to change anything in the cell structure or the cellular appearance on the blood smear. All right? What test is used to do to what test is traditionally done um, for PNH? We have what we call HAMS acidified serum test, and the confirmatory for that one is your what? Your sugar water test, okay? Or sucrose water test. How do we do HAMS acidified serum? You have three tubes. One, two, three tubes. Okay? First, your first tube is for cell suspension. Cell suspension. Other one is normal serum, or what we call an acidified serum. The next one is the acidified serum. How do we acidify a serum? We add 0.02%, 0.02% hydrochloric acid. Okay? Now, why do we need cell suspension? A 5% 5% cell suspension. We want to identify if the cell will lyse in acidified serum. And this one will serve as our control. Okay? So the normal serum, patient serum is here. You aliquot that patient serum here. Then you add the patient's cell suspension on both sides. Do we need to be specific? No. In the board exam, they're not going to ask you how many ml. They're just going to ask you what are the reagents. So sometimes they're going to ask you, acidifying a serum is responsible for, is useful for what particular disease? That's what they're going to ask you. Or using 0.02% HCl, what particular disease is being detected, right? Now, how do we do sucrose water test? Suppose that suppose that your cells don't lyse in this particular regard. All right. What you're going to do is you're actually going to activate the complements. So how are you going to activate the complements? Cells are already fixed with hydrochloric acid and they didn't lyse. Sugar water will actually, adding sugar water to this particular suspension will neutralize the acidity and allow the entry of glucose molecule, or sorry, sucrose molecules inside the cells, with that, which then will burst cells who have problems in the piga gene, with the piga gene, or cells without CD55 or CD59. Okay, so that's why it's confirmatory for PNH. All right. Is it still used today? No, not anymore. How do we test it? We do flow cytometry. Okay. So what acid are we using? The next question is hydrochloric acid, or HCl. How is PNH diagnosed today? Flow cytometry, detecting your CD55 and CD59. If you work in a laboratory in the Philippines, they're still doing Ham's acidified serum test. I remember that one because when I was working in the Philippines, we had a patient who had an idiopathic hemolytic anemia, and the doctors they didn't know how to diagnose the patient with normocytic anemia. The peripheral blood smear is that although the blood cells are small, they are lowered, the appearance of the cells in the peripheral blood smear are normocytic normochromic, meaning that there is no problem with the RBC indices. It just so happens that the anemia arises whenever on certain cases. So the doctors decided that we have to do a differential diagnosis. And the one thing that they can do is send it out for different hemolytic analysis testing. The first thing that they did was they do well, the first thing that they did was they asked for Coombs test. So Coombs test came out negative. Now they know that it's not an autoimmune hemolytic anemia. All right. What's the next step? The next step is finding which hemolytic anemia is causing the patient to have anemia. So they decided maybe it's paroxysmal nocturnal hemoglobinuria. So they asked us to send out the sample for Ham's acidified test, or they call it in UST hospital as acidified serum, acidified serum test. It's actually not Ham's acidified serum test in UST hospital, and they sent the samples there. I'm not sure if we actually did it, but the patient was diagnosed with paroxysmal nocturnal hemoglobinuria. That was his diagnosis, and how they did that was through acidified serum test and sugar water test as a confirmatory test. And after that, they sent the patient sample to, I think, Singapore. No, no, no. They sent it to St. Luke's Hospital, and the St. Luke's Hospital Research Center. They did flow cytometry on the patient sample and um, with CD55 only, and that's what happened to the patient's case. So they diagnosed it completely. So in diagnosing diseases, it's important for the laboratory people to understand the principles of the tests as well as the value of the tests. So if you aren't gonna learn a lot of things, if you're gonna learn something from this particular lecture, one thing that I can impart with you is the importance of the tests that we do, um, especially in diagnosing rare cases such as this. All right. All right. So true or false? PNH is a form of hemolytic anemia that occurs only at night. People in the chat, what do you think? True or false? I'll give you guys a minute to answer that question. Is it a form of hemolytic anemia that occurs only at night? True or false? People can say, you can type it on the chat box whilst I drink my water. All right. So most of the people answered false. All right. So why is that? Well, from a historical standpoint, in 1987, I think it's 1887, I think. 1882, sorry. 1882, it was discovered by the physicians discovered nocturnal hemoglobinuria. But because they don't have knowledge of complements that time, they thought that it actually happens at night. But in reality, it can occur at any time of the day. All right. But even though we already know that there's a problem with um, the complements and everything, we have knowledge about this one. Still, physicians use paroxysmal nocturnal hemoglobinuria as the term because it's they don't want to name it again. I suppose I think that's the only reason they don't want to name it again, because there's so much anemia that they have there, so much more so much anemia that they have to study, and they don't want to name it again. But that's the historical background for this particular disease. All right. Now, let's move on to G6PD deficiency. Does anybody remember their G6PD deficiency lectures in clinical chemistry? Why is it important in RBC, or specifically in the hemolytic anemias? Well, G6PD stands for glucose 6 phosphate dehydrogenase. All right, so it's a dehydrogenase substance. It's a dehydrogenase enzyme. Okay. So the major cause of anemia in G6PD deficiency is actually free radicals or oxidative stress, as we know it. And one way for the body to protect itself, sorry, RBCs to protect itself, because we all know that RBCs are enucleated. So when I say something is enucleated with the capital A, with the capital A, okay, enucleated, it doesn't have a nucleus. Remember last week we've discussed the actions, the process at which a particular cell will develop. Okay, so which particular cell am I talking about? I'm talking about the RBCs, of course, because that's the only cell in the body or in the blood vessels that doesn't have any nucleus. Um, One way to protect itself from lysis is through what we call the hexose monophosphate shunt. I'm not going to discuss the whole process because this isn't for um, medical students. This lecture is for more for medical technology students, and we just want you to guys, I just want you guys to understand the basic principles. So basically, what happens is that NAD, glucose 6-phosphate dehydrogenase. You're gonna look here, um, the hexose monophosphate shunt. This is what we call the hexose monophosphate pathway, which is responsible for removing free radicals. So the end of the whole process is removal of free radicals from reacting to your RBCs. So what happens is that NADP needs G6PD to become NADPH, glucose 6-phosphate dehydrogenase. Okay, so it transforms it into with the hydrogen group here. Sorry, let's remove that one. Eraser. So the hydrogen group here is added to that one by via your G6PD. Okay. Why does it need G6? Why do we need hydrogen for this one? Because you want to reduce glutathione. Oh, wow. Glutathione. Well, we're taking that one, right? Glutathione for whitening, right? But aside from that, if you can remember why how they market glutathione, it says you want to remove free radicals to protect the cells, blah blah blah. That's basically what the RBCs are doing. Removal of free radicals. Okay, so it has its own pathway inside it. It's actually a glycolytic pathway as opposed to a glycolysis is more of a anaerobic form of energy production. So in a way, it has to produce some sort of acid. Okay, and one way to remove those acids is with the use of glutathione and NADPH reacting with each other. When they react with each other, they will produce reduced glutathione, which can react with free radicals, and the free radicals will be dissolved. All right. Now, people with G6PD can't live normal lives. That's another true or false question. I want you guys to answer in the chat. I'll give you guys a minute whilst I drink some more water. 2 minutes. Try to challenge yourself, guys, without looking at the PowerPoint presentation. People with G6PD deficiency can't live normal lives. Yes, Earl. Why did you unmute your Earl? What's your answer? True or false? Ay, ignore ako ni Earl. False, 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 false. All right. Time's up. Correct answer is false. People with that disorder can live normal lives provided they avoid oxidative stress. Give me an example of a thing that we eat that has oxidative stress. Anybody know? There's a lot of things in the house, even in literature, that can cause oxidative stress. Anybody? Anybody knows? I'm gonna call people's names. What about Vanessa? Vanessa, hello. Oh, can you give me an example of something in your house that can give you oxidative stress? Chili beans? You're almost there, but you forgot. No, oxidative stress. I'm not talking about actual stress. Girl, I'm not talking about. Girl, you're so funny. Processed food, yeah, but it doesn't cause hemolytic anemia. Alcohol, no. Toxins, what specific toxin? Anything in your house. You put it in your cabinet. It's a white ball. Mothball, somebody already answered. Ella Villamar. What is mothballs made of, Ella? All right, thank you, Vanessa. Ella, where is it made out of? It's made out of naphthalene. All right, it's made out of naphthalene, and that can cause oxidative stress because there's a lot of free radicals in there. Inhalation of it can actually cause hemolytic anemia. I experienced one case of this, and when I was working in the Philippines, um, one of my colleagues was trying to prepare the baby's, they call this a baby bed or a cot for our extraction area. So we place the baby there. We usually clean it out in the morning, and the sheets are often stored underneath it because every shift we change it. Um, inside the cabinet, we sometimes put mothballs on that particular patient or on that particular location. And one day we were extracting blood from one of our patients, and suddenly the baby's arm turned red or some immediately had petechial dots on the left arm, and I noticed we noticed it was from the part of the body that's not away from that's within the bed cot. The arm freely moving away from the cot is not turning red with petechia. We immediately removed that patient from that place, and then we shared this particular incident to our supervisor, and we found out that that patient was actually G6PD positive, G6PD, G6PD deficiency positive. So it's confirmed. Usually in the Philippines, it's quite common. In the hospital that I work in, is actually we actually do confirmatory testing for that one. It's done in the clinical chemistry laboratory. But it's important for you guys to know the importance of mothballs because sometimes there are parents who don't get their children screened for newborn screening. There's a lot of tests that we do in the Philippines. There's actually a what do you call this, a newborn screening act, and although it's free in a lot of it's free and it's mandatory for a lot of the hospitals in the Philippines. Not many people still want to avail this particular test, and it's very important because it might kill a patient if you don't know that they have G6PD deficiency. All right, so that's a little story about G6PD. And I've encountered it firsthand, and it was a scary experience, especially when you don't know what's going on. Okay, so we're gonna move on with the drugs. So drugs can actually activate G6PD hemolysis, G6PD deficiency hemolysis. And again, this is another form of normocytic normochromic anemia. And the drugs that can cause is uh, we have a shortcut here, D6P. So what's the D6P? Sulfondapsone and Primequine. Why did I use six? We just want I want you guys to remember the letters. That's the reason why I added this one, D6P. This is the one that I can provide for you guys so that it's easy for you to remember this particular information. Okay, so why is it important? Is it important for you guys to remember it? Yes, of course. I'm not gonna put it as a mnemonic if it's not important. So I'm gonna put another star on it because you guys need to remember if it's okay, if it's high yield, so to speak. All right, sulfondapsone and primaquine. What's primaquine? Primaquine is a anti-malarial drug, so please don't forget that one. Okay? Trivia. What type of bean should be avoided by people with G6PD deficiency? All right. So anybody can answer this? Does anybody have any idea what type of beans? All right. Anybody knows? What about Christine Murillo? Beans. Hi, Christine. What type of beans? Hello. What type of beans should people? Nasabi na ba? Ay, hindi ako nakatingin sa chat. Ay, meron pala nagsagot. Oh my gosh. Active ang mga students ngayon. All right. Thank you, Christine, for speaking up. So, thank you sa pagbasa, Christine. Love you lots. All right. Love you lots, mama. Chup chup. Plutonic ha. Plutonic love lang, girl. All right. So, faba beans. So, if you don't know, this is what faba beans looks like. You have you have the one mixed with teriyaki sauce. The other one is dried. The other ones, I don't know why they're like that. But yeah, these are your faba beans. So please avoid this if you have G6PD deficiency. I added this as a trivia for you guys. All right. Denatured hemoglobin from oxidative stress results into what type of inclusion body? I think somebody already answered that one from last week's lecture, and I was pretty excited when he answered it in such a way. It's actually Heinz bodies. Okay, so if we says denatured hemoglobin, it's Heinz bodies. Our shortcut earlier, yesterday, last week rather, was Jolly D. Howell Jolly. D stands for DNA. Heinz bodies on this type of, for example, is denatured hemoglobin. He hemoglobin. So you guys don't get confused. Okay. Howell Jolly. Where's my pen? So you guys won't forget. Howell Jolly is D. Okay? DNA. All right? Howell Jolly. Okay? My shortcut is Jolly D, not Jolly B. Jolly D. So you guys won't forget. What about for Heinz bodies? For Heinz bodies, it is HE. HE, HE. What's HE? Hemoglobin. Denatured hemoglobin. So both of them are just basically remnants of DNA. The other one is a remnant of hemoglobin. All right. Laboratory findings that are common in patients with G6PD deficiency. We already know this. Coombs test. All right. Negative Coombs test. Normocytic normochromic anemia, Heinz bodies, and bite cells. Now I'm gonna give you an example. I'm gonna give you the reason why bite cells are present. Now, remember in our previous lecture, the action of the spleen in terms of hematopoiesis. I need someone to answer my question. Um, who here is available? Hmm. Let's check out the participants. Um, what about Anna Yusuf or Joseph? Anna, are you available? Yeah. Um, what is the action of the spleen in hematopoiesis? Anna, are you there? All right. Anybody in the chat can help Anna. Don't worry. You can chat. You can do whatever. Somebody's raising her hand. Pitting and calling. All right. I'm sorry, Anna, but somebody already answered for you. Hannah, how are you, Anna? Are you there, Anna? Hi, Anna. Oh no, Hannah. Sorry, not Anna. Hannah. Hi, Hannah. What do you mean by pitting and calling? Hmm, yes. Okay. Aside from that, what else? That's pitting. Okay. Culling is what? Removal of senescent RBCs, right, Hannah? Am I correct? All right. So damaged cells. Thank you so much, Hannah, and thank you for the other people who participated. Gina said filter. Filter is a very vague term, and I want you to be specific when you're answering. All right? I want specifics. It's okay if you can guess. Don't worry. Don't worry. We'll try to answer it correctly, so that you guys have an idea of what I'm talking about. All right. So in the spleen, I'm gonna make a drawing again. I like drawing right now. All right, so I'm gonna make a drawing right now. The spleen looks like a triangular. Yeah. So bite cells from. I think you guys already watched my video that I asked you to watch last week. So the spleen is like this. It's like basically triangular. It doesn't look like a spleen. I'm sorry, guys. So what the spleen does is inside the spleen you have what you call macrophages. All right. So macro. The macrophages has receptors that can detect the inside the sinusoids, sorry. So there's sinusoids inside, okay? Sinusoids. So inside the sinusoids, there's macrophages, and each macrophage will actually do the culling and pitting. So if it detects old cells or dying cells, probably damaged cells, what that does is it immediately kills it and it recycles whatever it is it has in whatever the RBC has left for recycling. Okay, whether it be the hemoglobin or the iron, that's what the macrophage will do. Okay, so if a cell comes into the spleen where there is Heinz bodies, for example, which is denatured hemoglobin, it will actually take a bite out of the cell, and the cell would look like this in the peripheral blood smear. Imagine you're eating a donut and you took a bite out of it. Look at that donut. It has the markings of your mouth on it, and that's basically what happens to the cells. And that's the reason why they're called bite cells. So remember, in G6PD you will also find bite cells. Okay, because there is a denatured hemoglobin precipitate on the cell surface. All right. So why is it again Coombs test negative? Negative for Coombs test. It's because it's not autoimmune. All right. Okay. So patients with G6PD have been known to have a resistance to what type of parasites? Um, what parasite? Plasmodium falciparum. It's gonna be asked again in blood banking. So I might as well put it here so you guys remember. Now we move on to the next chapter of our hemolytic anemia, or rather, um, normocytic normochromic anemia, but in this case, it's a little bit microcytic. So I would say it's from microcytic. It's like a cell from, yeah, but that's for the latter cases. But anyway, I would classify this as normocytic normochromic. Now, the main function or the abnormality in these types of patients is that they don't have a normal skeletal structure in the RBC. Is there skeletal structures? Yes, sir. There is the skeletal structures, the skeletal proteins. That's the one that we're talking about. So there's three types of skeletal proteins that you're going to have to talk about. This is ankyrin, another one is spectrin, and the bands. So there's a lot of bands that we have to talk about. So I just put it as bands. Okay, never encountered an exam where it asks the specific band. Okay, because they have different functions, and I don't think it's important for the board exam to specify what specific band. So I just wrote in here bands. What is important is that they're going to ask you about spectrin or ankyrin. So guys, please remember that the abnormality for patients with hereditary spherocytosis is this one, ankyrin and spectrin. Okay, how can I remember that one, sir? Well, anchoring sounds like an anchor, so it anchors the shape of your RBC. Spectrin is where you spectate. I know I'm just making neural connections for you guys. So I guess what do you call this? My mnemonic for this one is that you're going to look for a boat that has an anchor that's anchored, and the one up there is somebody who's spectating. So in cases of hereditary spherocytosis, there's no spectator and there's no anchor. That's my that's my mnemonic for you guys. So please don't forget that's the dysfunction for patients with hereditary spherocytosis. So what happens to the shape of the RBC? Basically, I've given you guys an example of this one already. So you guys already know that they're shaped as globe-like or sphere instead of being biconcave. And the reason why it's considered to be another form of hemolytic anemia that can cause splenomegaly is because of the size and the shape of the cells, they can't squeeze in the sinusoids, and therefore they lodge into organs such as the spleen. All right? So that results into splenomegaly. All right? Usual laboratory findings for patients with hereditary spherocytosis. Because it's a hemolytic anemia, we're going to test it again for Coombs test, and it's negative again. And it's normocytic, microcytic, and one thing that you need to remember is that it's hyperchromic. All right. Why is it hyperchromic? Because as I mentioned before, they're globe-like. They don't have a central pallor. All right. They don't have central pallor. And another thing that you can find about that you can think about them is Howell Jolly bodies. What is Howell Jolly bodies made out of? DNA remnants. All right, DNA remnants. So please don't forget that one. Why is there DNA remnants? I thought that happens only in patients with um, with DNA abnormalities or cell what they call this cell division cell multiplication or cell division properties. Why does it have Howell Jolly bodies? Well, because of the shape and the rapid production of the cells in this because it's a hemolytic anemia, the cells itself are rushed to go out of the bloodstream. That's why they suffer from abnormality in shape as well as dysfunctionality. And what happens to them is that the DNA from being rushed to go outside the bone marrow, the DNA remnants get left behind and they get stained. And that's the Howell Jolly bodies. Bilirubinemia, what happens? Why is there bilirubinemia in these kinds of patients? Because there is splenomegaly, and the cells themselves, they try to get stuck on the spleen, and what happens is that they just pop in the spleen and they get released into the blood. The substances that they have inside the cells get released into the bloodstream, and that one of those is the unconjugated bilirubinemia. All right. So what's the reason why they're present? Because the RBC macrophages cannot remove these RBC inclusions. So that's the reason why. Okay, because of the shape. Okay. Aside from that, aside from that, they are rushed to go outside the bone marrow. All right. What's the gold standard test for the diagnosis of hereditary spherocytosis? This is your osmotic fragility test. Okay. Osmotic fragility test is described in the details of the osmotic fragility test is described in my previous lecture. If you have the copy of that one, think that's lecture number 3, the one with the black background. That's where that's where the complete discussion of osmotic fragility test is. But I just we'll just go on a quick uh, discussion on this one, specifically the methods because it's important. So we have what you call the Sanford method and Dacey's method. Sanford method is more on visual observation, while Dacey's is much more quantitative because it uses a spectrophotometer. All right. So what is the OFT results for hereditary spherocytosis? There is a decrease in, there's no answer. What happened? Oh gosh. Oh my god. All right. Spherocytosis. Why is there no answer? All right, we're gonna make another one. So the OFT result of patients with hereditary spherocytosis is actually increased. Okay, increased. What do we mean by increased? It means that it easily gets the cells get easily hemolyzed. Okay, all right. What sample is used for patients with OF for to be who are to be tested for OFT? We use defibrinized blood. How do you make defibrinized blood? Basically, you put blood in a test tube with metal beads in it, or glass beads rather, and then you shake the glass beads. And as the fibrin is being caught on the glass beads, the sound of the glass beads hitting the bottom of the test tubes will diminish, and a, a fully defibrinized blood sound has no sound whenever you move the even when there's the presence of glass beads at the bottom. So that's how we defibrinate it, manual defibrination. All right. So what reagent is used in OFT? Okay. So what reagent is used in OFT? Well, we use isotonic solution of NaCl or sodium fluoride. That's 0.5% NaCl. So we just dilute it. Okay. RBCs with patients with hereditary cirrhosis easily lies in hypotonic solutions. All right. And what cells can resist hypertonic lysis is your sickle cells, okay, or codocytes. What are codocytes? These are cells that are considered to look like Mexican hat cells. Okay. Now, I think this is the last topic for anemias. Oh no, there's more. I think we're going to discuss aplastic anemias after this, but yeah, we're gonna move on with macrocytic anemias, and again, I'm gonna go and give you guys like this time 15 minutes of break before we discuss macrocytic anemias. All right, is that okay? 15 minutes this time. Hel- All right, all right. Very attentive when I'm talking about breaks. So 15 minutes starts now.